This protocol allows for the non-destructive imaging of intact Drosophila at micron resolution. It can be used to understand mechanisms of animal physiology, anatomy, and development. Because the flies are imaged in an intact state without the need for tissue dissection, the spatial architecture of tissue and organs are maintained in their native state and are easily observed. MicroCT is an ideal tool for studies aimed at modeling human diseases in flies or for understanding developmental defects that affect quality of life. It has also proven to be useful in other disciplines such as engineering, physics, material science, geology, and anthropology.
After selecting the developmental time point of interest for imaging, transfer 5 to 20 animals into 1 milliliter of PBST in a 1.5 milliliter microcentrifuge tube, and incubate the tube for 5 minutes at room temperature with periodic tapping to assist the removal of hydrophobic coating and allow the animals to become fully submerged. At the end of the incubation, replace the PBST with 1 milliliter of Bouan's solution and tap the tube to fully submerge the flies. For larval and pupal samples, after a 2-hour incubation at room temperature, wash the animals 3 times for 5 minutes and 1 milliliter of fresh PBS per wash. After the last wash, transfer the samples to a multi-well dissecting dish containing fresh PBS per well under a dissecting microscope and use a small minution pin attached to a holder to poke a hole in the anterior and posterior cuticle of each sample, taking care